This tutorial will require a Google Cloud account, and our search on BigQuery will cost a few cents. If you don't already have a Google Cloud account, sign up so you can follow along. It comes with $300 in free credits, so you won't need to spend any actual money. Often, the hardest part of working with cancer data isn't doing the analysis, it's just finding and combining the data in the first place. Genomic data, proteomic data, imaging data can all be spread across different repositories in different systems with different formats and different standards. In this video, I will show you how tools like Cancer Data Aggregator and ISB-CGC let you search across systems, locate overlapping data, and build a usable cohort. So we will build a query, combine that data into a single data set, and do a small analysis. To get started, we're going to go to the Cancer Data Aggregator website, which is cda.readthedocs.io. And on the home page, you'll find a button that says Launch CDA in the Cloud. We're going to click that, and it will take us to a Google Colab site. So here are all of the example notebooks created by CDA and others that show you how to search and use Cancer Data Aggregator data. If we go to the bottom one, you'll see our newest one, which is using CDA data with the ISB-CGC BigQuery tables. We're going to click that, and it will open up the specific IPython notebook that has the code for doing this analysis. Now, because this is on Google Colab, all of the code is pre-written for you to do the analysis that I'm going to show you today, but you can also edit it, so you can use this as a nice starting basis for your own analysis. In this example, we're going to look for subjects that have mutation and gene expression data, protein intensity data, and slide images. And then we're going to show you how to use that list of subjects to pull the actual data from ISB-CGC and feed into a simple analysis. You can imagine that this would be really useful for doing things like seeing whether a mutation correlates with a particular amount of gene expression or protein intensity, or whether you can see differences between images that do or do not have a specific mutation. To get started, we have to set up our notebook. To do that, in an IPython notebook, we hit the play button. The first time that you do this, you will get a warning that it wasn't authored by Google because it was authored by CDA, so hit run anyway. You'll notice that you get a little check mark after a few seconds that says that this code has run completely and is fine. When you run this, it will take something more like 15 seconds the first time that you install all of these things. But I have pre-installed it to save us some time. Next, I'm going to import all of the different functions from the CDA Python tool we just installed so that we can use them directly in this notebook. Now we can really get started. What we want to do is discover data that we don't already know about. So we aren't going to tell CDA Python that we want a certain set of individuals, we're going to discover them. To do that, we need to know a little bit about the data that is at CDA. So we're going to hit play on this box that says tables, and what it shows us is what tables are available in the CDA database: file, mutation, observation, project, subject, treatment, and upstream identifiers. Then we would pick a table that we think that we're interested in. Here, we know that we're looking for all of these different file types, so let's look at the file table. I want to know what columns are in the file table so that I have an idea of what kinds of data are in there. So again, I hit play, and I get the list of every column in the file table, along with a description of what kind of data is in each of those columns. And remember that there can be multiple pages. Let's see if the values that we're looking for appear in file type. And to do that, I'm going to say column values. What are the column values for file type? And here, I am going to get a whole bunch of answers because the CDA database is very, very large. So you can see that we have over 300,000 somatic mutation indices. We have lots of CT images. We have lots of aligned reads. And here we have annotated somatic mutations. That's one of the ones that we were looking for. We also wanted to look for slide images. So if I click up here and do a search, I can see that there is a slide image available. There's about 35,000. And we can do that for each of the file types to see how much data is available. Once we're satisfied that all of our different values are in here, we can start to run queries. But before we run a query, let's look at the syntax for how one does that. This is just a short example of what it looks like when you run CDA Python. So let's say that we are interested in pathology reports. We would say that we want our file type to equal pathology report. And because that is an important thing we're telling the computer, we put it in quotes. We are setting that inside of a bracket that's labeled match all. So we could put one thing in this set of brackets or 50 things in this set of brackets. And either way, it would match every single thing that is inside of match all. That is nested inside of this function called getFileData, because again, what we're interested is getting those files so that we can see what the pathology reports are. If I set all of this in the direction the computer's going to read it, it's going to go get file data that matches all of these characteristics, where the characteristics are that file type equals a pathology report. And I am now saving that information as a variable called pathology, and then I'm only going to have it show me the first 3 rows of pathology when it's done, because I'm probably going to get lots and lots of answers. So if I hit play, this will take just a second to run. And you can see that I get back 3 rows of pathology reports. These all happen to be from GDC. They all happen to be about tumors. They all happen to be PDFs. But we can also see that they're all open access, so we should be able to get to any of them. And there's a DRS URL. So this is a direct link to those files. You can put this into a resolver service, and it will take you directly to where these files live. Now that we've gotten ourselves familiar with the basic CDA syntax, let's start building the cohort that we're interested in, the one where subjects have genomic, proteomic, and imaging data. So recall that the genomic data that we were interested in was somatic mutations. So just like before, we have to tell CDA Python what kind of information we're interested in and what column to look for it. These are file type, and it happens that somatic mutations can come as either aggregated or annotated. We're going to put both of those inside of the quotations and inside of the little brackets. But here, instead of using match all, we're going to use match any. That's because we don't need a person to have both an aggregated somatic mutation and an annotated somatic mutation. We just need one or the other. And since now we're looking for people, instead of looking for files like we were with our pathology example, we're going to get subject data. We're going to save this as a variable called mutation, and just like before, we're just going to look at the first 3 rows to make sure that our data looks like the way that we wanted it to. So when I hit play, CDA is going to go and find us all of the subjects that have one of these file types. Here are the first 3. All of them come from GDC. That's to be expected. The Genomic Data Commons holds the vast majority of the genomic data that's available at the National Cancer Institute. These are all from humans, and they all have similar subject IDs, so these 3 were probably from the same project. But we can see that these 3 people have both aggregated somatic mutations and annotated somatic mutations. So this looks like the data that we were expecting. Let's go see what we can get for gene expression data. Now, here we're doing the same thing, except we want everyone to have gene expression quantification as their file type. So we're going to do match all and get subject data. Here, I have the first 3 subjects. These are also all human. Mostly this data comes from GDC, but the first person also has some IDC data, and they all have gene expression quantification files. Now, let's look for measures of protein abundance. This will probably all come from the Proteomic Data Commons, because that's where most proteomic data lives. So we're going to, again, do file type. We're going to look for masked intensities this time, match them all, get the subject data, and save it to a variable called intensity. And here we are. We have 3 humans. We have their data sources, and most of these subjects have data at multiple different DCs, but they all have masked identity file types, so that's good. And then finally, we're going to look for those slide images. Because they're images, they'll probably mostly be from the imaging data commons, but there are images in a few other places as well. So file type equals slide image, and let's get that subject data. We have our 3 people that are at the top of this list. All of them have slide images, which is good. And for these first 3 at least, they all have data at multiple places. So now we have all of our different variables that have the subjects that have slide data, that have various genomic data, and the expression data. The first thing that we can do is we can look at a list of the people that have all of these types. We did each one individually, but we didn't look to see yet whether there is overlap. An easy way to look for overlap is to do a Venn diagram. Since we only have 4 options, it should be an easy one to visualize. There are ways to use CDA Python to check for overlap, but an easy visual way is to make a Venn diagram. So let's do that instead. First, we're going to install this Venn diagram tool, and at the same time, we're going to import the big query tools that we need to go and get the data from ISB-CGC in a couple of minutes. To make the Venn diagram, we have to give it the sets. Our sets are going to be easy because we have 4 different data frames that are the results from our 4 queries. So we'll have a mutation set, an expression set, an intensity set, and a slide set, and each of those will be made from the subject ID of the corresponding data that we've already queried from CDA. And then we're going to plot that as a Venn diagram. So when I hit play, it will go grab all of the data we've already pulled from CDA, pull out the subject IDs, and then see how they overlap. Now, since we only have 4, this makes a pretty nice diagram, and we can see that there's a little bit over 10,000 subjects that had data from all 4 of those different areas. So since we've got the IDs pulled out already, we are going to make a set that only tells us what those 10,000 or so IDs are that we could feed into ISB-CGC. So our final set is going to take the intersection of all of those places, and then we're going to format the identifiers in the way that we need to put them into ISB-CGC. And you can see that it has just made us a list of all of the identifiers in a comma-separated way. Note that we called this list formatted identifiers. We're going to need that again when we get to ISB-CGC. But for the moment, let's talk about how ISB-CGC works. ISB-CGC is in BigQuery, which is a Google search tool. And the data is separated into a whole bunch of different tables in BigQuery, each with different kinds of information in them. You can go to portal.isb-cgc.org to see a list of all of the tables that are made available in BigQuery by ISB-CGC. And if we went through all of those, we would see that there are 5 different tables that we are interested in. There is the one from TCGA called versioned RNA-seq. There is one called Somatic Mutation from TCGA. And then there are 3 that are for proteomic data. And these are for breast cancer and ovarian cancer. If you're interested in other diseases, you might want other tables. But for this example, we're going to use these. Google does charge a small amount for running queries on BigQuery. And you will need an account on the Google Cloud. If you don't have an account on Google Cloud, check the links below for instructions on how to get one. Since Google Cloud charges a small amount for each search, you will have to tell it what project it should charge. So here, in this box, you'll need to change your Google project to the name of your own Google project. Remember, this will only work for one that you have an actual account on. So if you just try to use mine, it won't run. Make sure that you put in your own Google project and hit play. It came back with a check mark, which means I am now authenticated and this should work. So let's run some big SQL queries. Big SQL queries are a lot like using Excel macros or subsetting data frames. You might be familiar with a syntax that looks something like this. Some data frame, use this column, and meet this condition. In SQL, what you would say is select some column from some table where it meets some conditional. First, we're going to look for the FPKM values for the RNA sequencing and the gene names, and we're going to use that big list of identifiers from CDA to pull out only the subjects that have the characteristics we want. And for this moment, we're going to focus on the gene ERBB2. We package this query up in a variable called expression query, and then we use the client query tool from Google Cloud to send it to BigQuery. And just like before, we're asking it to only give back the first 3 lines. So our query will be select some columns. Here we're selecting 5 of them from some table, and this one is the RNA-seq HG38. In the first 3 results, we have 3 different case barcodes, all from the same project. They all, of course, have the gene ERBB2 because we set that in our query, but now we also have the FPKM for all of our subjects. Now we're going to do a similar thing from the mutation table. So we're going to select the case barcode, the project short name, the symbol, and the variant classification from a different ISBCG table, the Somatic mutation table. Look for only the cases that are in our formatted identifiers list, and of course, we're going to keep the ERBB2. When we send that query, we get back the first 3 lines, and we can see that these case barcodes have different variant classifications. Now, let's take our proteomic data and do a similar thing. Here we're going to select the case submitter ID, gene symbol, the protein abundance, which is the thing we actually care about, and the sample type of these aliquots. And this is a much more complicated query because we're going to take multiple tables from ISBCGC and search all of them at once. So here, we're going to search the proteome TCGA breast cancer table, the quant proteome ovarian cancer table from this particular project, ovarian cancer from the PNNL project, and join that all together into a single search. We're going to search on the case IDs, and of course, we're going to filter by the identifier list we already have, and for gene ERBB2. Let's send that to Google and see what the first 3 results look like. So now we have the protein abundance for ERBB2 and the case IDs so that we can combine these data sets back together for our subjects. Now we're actually going to use BigQuery to join all of these together rather than doing something manual. So we're going to combine our query by selecting the barcodes, the project names, the gene names, the FPKM, the protein abundance, and the variant classification. And we're going to select those from the various tables of answers we've already gotten, our expression query, our abundance query, and our mutation query. And we're going to join all of those so that we get only the subjects back that had data in all of these. So when we hit play, we should get back a big combined data frame. And here, I've just asked for combined data frame, not the first 3, so we can see all of them. So here we have 248 subjects that had all of those data types available at CDA, and we were able to find them in the ISBCGC databases. ISBCGC doesn't have all of the files that CDA indexes, so we would expect for it to be a smaller sample than we got for our overlap at CDA. But you can see that now we have all of the different FPKMs, the protein abundance, and the variants in one table. So this is really convenient and enough that we can start to do some simple analyses. Let's just do a simple plot where we're going to compare the log FPKM to the protein abundance. This is the sort of thing that you would do when you are first looking at your data to see whether it has the characteristics that you need to do your further work. So we are getting the log of the PKFM, and then we are plotting it against the protein abundance in log 2. As you can see here, we get a pretty reasonable distribution where protein abundance is basically a function of the FPKM. We can also do other simple analyses. For instance, we could look to see whether the RNA expression is different between different mutation types. So we could take our variant classifications and pull out all of the missense mutations and all of the not missense mutations and plot those against each other. So now we have missense versus wild type. And when I run that, I get a box plot that shows me that the missense mutations tend to have a log FPKM around 4, but the other ones have a much wider spread and a lower average. So this might be really interesting to look into later because it looks like the missense have a much narrower range of protein abundance values. So in the last few minutes, we have used CDA to find a large cohort of subjects that have overlapping data from imaging, proteomics, and genomics. And then we have used those to query against the ISB-CGC, which has real data for some of these subjects. And we've used that data to do some initial exploratory analysis. At this point, you could start asking real questions, like how gene expression relates to the protein abundance, or how mutations affect both. Or you could bring in the imaging data and see what correlations are there. What makes this workflow powerful isn't just the code, it's the tools behind it. The Cancer Data Aggregator let us discover and locate data from across systems. We found subjects from the imaging data commons, the proteomic data commons, and the genomic data commons without having to go to any of their sites or learn any of their models or dictionaries. And we used ISB-CGC to pull out some of that data and analyze it without having to move any data to our local computer. Together, ISB-CGC and CDA have removed one of the biggest barriers to multi-omics research, just finding and aligning this data in the first place. If this was helpful, feel free to like and subscribe, and check out the notebook that we're using to try it yourself. If you want more tutorials like this, let me know what you would like to see next. And if you have any questions, please visit us at our help desk. The link is going to be in the notes.